quality control test. Quality control test means evaluation of your aerosol. Now quality control test consists of evaluation or the testing of your initially propellants, testing of your valve, testing of your actuators, and testing of your dip tubes. Then testing of your container, whatever the material we are using, that material we have to test for the higher pressure also, for the any kind of reaction purpose also. Then we have to check the weight, we have to check for the leakage purpose, because internally there is a high pressure. So sometimes after the finishing, after the total production, when we are transporting your material, there might be chances of leakage. So your container should not be having any kind of leakage. Then spray pattern, what kind of spray pattern is there, that also you have to test. So these are the quality control tests. Basically we have to carry out them for the your components of the your aerosol, that is for the propellant, for the valve, for the container, for the your wet purpose, for the leakage purpose, and for the spray pattern purpose. Now when you are selected your propellant, so basically we have to go for the identification of that propellant. It means what kind of propellant we have chosen, whether it is properly we have selected that propellant or not. So that can be tested by the gas chromatography as well as the IR spectroscopy. So here we can identify that propellant, which is that propellant, and what is the purity of that propellant. So purity, acceptability, as well as the identification test we have to carry out for the propellant. Then whatever the spray pattern developing devices is that is valve, actuator, and dip tube is there. So it is responsible to deliver your drug or to deliver your product from the container to the your selected target area. So that valve, actuator we have to check for their quality. Generally testing of that valve, testing of actuator, and testing of that dip tube is carried out by using the various kind of standards. So that standard are called as the military standards. So as per the military standards, we have to go for the testing of your valve, testing of your actuator. Because suppose we have prepared your aerosol, we have developed properly your all the product concentrate. So after that, suppose your valve or your actuator is not having the proper action, then your spray pattern will not be proper, your drug will not be delivered proper. So that's why whatever the valves, whatever the actuators you are using and container you are using, that should be tested for their quality. Generally these military standards are developed by the three kind of committees. First is the Aerosol Specification Committee, another is the Industrial Pharmaceutical Technology Section, another one is the Academy of Pharmaceutical Sciences. So these three bodies have developed your military standards. In short, they are called as the MIL SCD 105D. So for testing of your valve, testing of your dip tube, so we have to use some specific type of reference or the standard solutions. They are called as the test solution. So to check your ability of your valve, ability of your test tube, dip tube, so first of all we have to take the one particular container, and in that container we have to add your test solutions. Now test solutions are prepared by adding the various kind of mixtures. Now ingredient of test solutions, there are the three type of test solution are available for testing your ability of valve as well as the dip tube. Now test solution A is containing the isopropyl myristate 0.10%, then dichlorofluoromethane 49.95%, then tetrafluoroethane 49.95%, then trichloromonofluoro, then alcohol and specific gravity. So these all the contents are there for the test A, and then for the test B and test C, ingredients are same but the concentration is varied. So any one solution we can choose for the testing of your valve as well as the dip tube. So how to test? First of all, you have to take the 25 containers, and in that we have to add your test solution A, and then we have to fix your selected wall as well as the dip tube. Then after fixing that assembly, we have to expel your component just by pressing your wall for the 2 seconds. Suppose you have filled your test solution inside your container and you have attached your wall plus dip tube and you have pressed it for the 2 seconds, then you have to take the weight. So after the filling of your component, suppose you have taken the initial weight, and then after pressing your wall for the 2 seconds, we have to take the again weight. It means how much content from that container is delivered after pressing 2 seconds, we have to note out. Then the same procedure we have to carry out 2 times. It means first time we have pressed for the 2 seconds and some content is delivered. Note down the weight of that container. You will get the idea that from the initial amount, how much amount is delivered when you are pressing first time for the 2 seconds. Again we have to press your button and we have to press it for the 2 seconds, and we have to note down that how much amount is delivered. So initially we have taken the weight of your bottle containing your all the containers. Again you have pressed your wall for the 2 seconds, again some content is delivered, then we have taken the weight. Again second time you have pressed it for the 2 seconds and you have taken the weight. So likewise we have to carry out this procedure for all the 25 containers, and then we have to note down the ability of that wall at which manner they have delivered the drug. Now into the 25 your containers, that all the containers should deliver the same amount of product if you are pressing it for the 2 seconds for the 2 times. So for each container we have to note down it, and then we have to carry out some calculations. Now that calculation is that individually delivery of the amount of that material into the milligrams. It means when we are pressing it for the 2 times, how much amount is expelled from that wall. So that we have to note down, and we have to divide it by the specific gravity of that solution. Then it will give you the idea that what is the ability of that wall to deliver the amount from your container. So in microliters you will get the amount which is delivered by your container from that wall. So that wall's capacity will be calculated in the form of microliter. Now there are some limits. For each wall we have to calculate such kind of value. In microliter you will get the idea that how much amount of microliter can be delivered by that wall. So by this way we can carry out this test. Now in this case there are some limits they have given. Suppose your device have delivered the 54 microliter or less than 54 microliter. So in that case there is the 15% plus minus is we can say permissible. Sometimes it may deliver the 54% plus minus 50%, 15% more or the less. Another limit is there, that is for 55 to 200 microliter. Sometimes some wall is having a high capacity, they can deliver minimum 55 microliter or they can deliver the up to 200 microliter. So in that, suppose your wall is having that ability to deliver the 55 to 200 microliter. So in that case 10% limit is permissible. Sometimes 10% may more delivery or less delivery may be possible. So suppose your wall is having the you can say small capacity, it is having the ability to deliver the maximum 54 microliter amount or less than 54 microliter amount. So in that case we can have the 15% we can say concession or the permission. So in that case it may pass. Suppose your such kind of walls are not obeying these rules, or there is a we can say your limit is crossing, then again 50 walls you have to take and again we have to check it by this way. Suppose you have taken another 50 walls and you have tested it. So for that purpose there are some another limits. Suppose 4 or the more than 4 walls of your container or your selected walls are having the, we can say, outside the given limit, then your walls should be rejected. Suppose 3 walls deliveries are outside of the limit, then another 25 walls we have to test. It means if your first 25 walls are outside out of the limit, then we have taken suppose 50 walls. Again 50 walls suppose you have tested, and among that suppose 3 walls are outside of the limit. It means again we have to take the extra 25 walls and again we have to test. Suppose after that, in the last case or into the third stage, suppose from that 25 walls, suppose one wall is outside the limit, then we have to reject all the walls. If the 2 deliveries from the wall are beyond the limit, then another 25 wall we have to again test. So these are the specifications given for the testing of your walls. Now the containers. Suppose you have prepared your containers. Suppose you have chosen metallic or your glass type of container or the aluminum type of container is there. So first of all we have to examine that container for any kind of defect, because your containers which are prepared by the metallic, generally we are using the welding or the soldering procedure. So in that case some leakage may be there or they may not properly welded. So we have to check them with the naked eyes, as well as we have to check them for the conductivity of the electricity current, and then we have to check the glass containers also for the any kind of cracks inside your container. Then weight checking. Generally weight checking is done by periodically adding to the filling line tiered empty containers. So this weight checking is carried out. First of all, empty containers weight we have to take, and again we have to fill up it with your required amount of material, and then again we have to weight. Then what is the actual weight of your product is there. Then again we have to remove your container content from the container, and again we have to reweight. So by this way we have to check the weight of your product. It means without product how much weight is there, and when we are adding the product how much weight is there, and when we are removing the water your content, then at that time what is its weight. So by this way we have to check the weight of your actual product. Then leak testing. Generally after filling there might be chances that your wall is not properly fixed. It may have the leakage, so your gasket may not properly fix, or your container wall may having the small cracks. So that kind of problems may cause the leakages inside your container after the packaging. So that kind of leakages we have to check. Generally such kind of leakage testing can be easily done. Just we have to fill up your container and keep it inside the water bath, and then we can observe your leakage. Then spray pattern or the spray testing. So this spray pattern is very easy. We have to check what is the spray pattern is there after the pressing your wall. So such kind of spray testing can be carried out by using the we can say just we have to spray it onto your just pressing your wall, and we can check that how much area is covering, what is the spray pattern, how much size of the droplet is there. So such kind of spray testing we have to carry out. Now evaluation test. So after that we have to go for the evaluation of your actual product. Suppose we have prepared your aerosol containing your all the active ingredients as well as your all the propellant, and we have fixed your wall as well as deep tube. Then suppose you want to sell it into the market. Prior the selling we have to go for the evaluation. So here already we have discussed some tests. So that test was basic for the selection of your valve, for the selection of the dip tube, and selection of the metallic or the glass type of material for the preparation of the container. Now suppose we have prepared your aerosol and filled inside your container. Now we have to go for the another evaluation of that total container along with your product. Now first test is called as the flammability test and combustibility test. Under this flammability test, we are judging whether your product is having ability to catch the fire or not. Then when suppose we are pressing your material and it is producing the spray, and suppose at the same time there is any flame, then how much long that flame will be projected, that we have to check. So these total two properties coming under the flammability and combustibility property. As your propellant is having the ability to catch the flame, that's why we have to check your aerosol container for the flammability as well as the combustibility property. Then another property is the physiological characteristics of your aerosol container. Under the physiological characteristics, we have to check how much pressure is there inside your container, then density of your total content along with your propellant plus your mixture, and then moisture content inside your propellant, then identification of the propellant. Identification of propellant already we have seen. Then performance of your total aerosol container. Under the performance, we have to check the aerosol wall discharge rate. It means at which rate your product will be delivered to the patient, or when we are pressing the wall, how much amount of content will be expelled by your wall, that we have to check. Then spray pattern, what is the spray pattern is there. Then dosage with meter walls. Suppose we are using the meter wall, then how much amount of dose is expelled by your meter wall, whether it is more or less after your adjustment. So that also you have to check. Then what is the net content of your total container. Then what is the foam stability. Suppose you are using the foam system, then whether your foam is producing or not. Then suppose you are using your suspension type of your aerosol, then whether it is having the ability to deliver that material or not. What is the particle size of the drug in case of the suspension. Then biological testing we have to carry out. Under the biological testing, we have to check the therapeutic activity of your aerosol content, whether it is having the ability to give the therapeutic effect or not. Then toxicity studies are there. Sometimes in the disadvantages we have seen that there is a toxicity is may possible into the aerosol. So whether it is having the ability to give the toxic dose or not, or whether it is safe. So biological testing we have to carry out. So see, these are the 4 type of tests are there as the evaluation test for your aerosol. Now the first test is the flammability and combustibility test. How this test is carried out? This is flammability test. Generally under the flammability and combustibility there is a flash point test. Under the flash point test, what we have to do, there is one apparatus that is called as the tag open cup apparatus. So by using this apparatus we can carry out your flash point test. Now here initially for conducting this test, first of all, whatever the aerosol you have taken for the testing purpose, first of all just reduce the temperature of that aerosol or chill it by adding it into the refrigerator. Now when we are chilling it, whatever the gas is present inside your aerosol container, it will convert it into the liquid state. Now take some amount of content of that aerosol, just press it and you take some amount of that content. Now when we are taking the liquid from your container, initially it will be very chilled or it is in the liquid state. Now keep it on your apparatus. That apparatus is called as the tag open cup apparatus. Now slowly increase the temperature. Now this apparatus is having one facility to note down the temperature, to note down we can say to give the temperature also, or to provide the heat. when we are keeping the some amount of content of aerosol inside that container, slowly it will heat, and after a particular period what will happen, your material will cause the we can say develop the flame, or it will cause the burning. So at what temperature it has caused the, we can say, burning, or it has developed the vapors or ignition. So that ignition point is noted down inside that apparatus. So that temperature at which your product's vapors will get ignite, or it is developing the flame, so that point is called as the flash point. So simply, when you are using your aerosol at the home also, so suppose because of your outer temperature your aerosol is getting heated. Now suppose you want to use that aerosol and just you are pressing it. So because of the environmental condition, sometimes it may cause the development of the ignition or the flame. So to avoid such kind of situations, we should have the idea that your content at what temperature is having the ignition point or the ignition temperature. So that kind of temperature we can note down by using your flash point test. So simply take the content, heat it, and at which temperature it is causing the flame, or it is igniting. So that temperature is called as the flash point of your material. So this test is carried out by using the tag open cup apparatus. Now another test is called as the flame projection test. Now in this process or in this test, simply spray the product for the 4 seconds into the open flame. Just we have to take the one flame or any lamp is there, we have to take, and on that lamp we have to spray your aerosol for the 4 seconds. When we are spraying for the 4 seconds, immediately it will catch your flame and it will get we can say ignite. And then you just note down at how much long that spray will be there along with the flame. So that length of that flame we have to calculate into the, we can say we have to note down with the help of ruler, and into the centimeters how much long is there we have to calculate. So that is called as the flame projection. So it will give you the idea that suppose any patient is using your aerosol, and suppose nearby there is any lamp which is already ignited. So whether your aerosol will catch that flame or not, that can be you can say note down by using this flame projection test. Sometimes into the villages, some people are using the lamps, and there are some patients inside their home, they are using your aerosol. So accidentally they may catch your flame. So to avoid that, at what distance your aerosol are safe in such kind of homes there is a lamp or the open fire is there. So we can maintain such kind of distance, or we can say to the patient that up to a particular distance we should use such kind of your aerosol. Now physiological characteristics are there, that is vapor pressure, density, moisture, as well as identification of propellant. Your vapor pressure can be calculated by using the pressure gauge, or we can use the can puncturing devices for using your vapor pressure. Then your density of your product is calculated by using the hygrometer or the cyclometer. Then moisture content is calculated or find out by using the Carl-Fisher apparatus or the gas chromatography. Then identification of the propellant is done by using the gas chromatography or the IR spectroscopy. Then performance test. Under the performance test, we have to check the aerosol wall discharge rate. What is the aerosol wall discharge rate is there? So we have to carry out. Simply you take any kind of filled aerosol container and press it for the 2 seconds. Now initially prior to your pressing, you have to take the initial weight of your product, and after pressing you have to take the weight of your product. and then find out that how much amount of product has been expelled after the pressing. So by this way we can take the your discharge rate of your wall. Then spray pattern. When we are pressing your wall, at what is the spray pattern is there, whether it is elongated or whether it is broad, such kind of spray pattern we have to find out. For that purpose, there is one specific type of paper we can use, which is already treated with the dye talc mixture. So here, when we are pressing your wall, it will give the spray and it will strike to the that paper. So depending on the velocity of that large particle and low particles size, so depending on their velocity, they will spread on that paper, and then we can note down your spray pattern. Now another property is the dosage with the meter walls. Dosage with meter wall means when for the particular type of aerosols we are using the metered walls, it means they are having the ability to measure the dose of that content. So in that case, suppose we are using such kind of meter walls, whether they are having the ability to expel accurate quantity of material or not, that we have to check. So for that purpose we have to go for the testing of that meter walls. So here we have to take your container along with your meter wall, which is already fixed. Then we have to first of all we have to take the weight of that material, and then total containers weight we have to note down, and then we have to just press your meter wall, and then we have to take the another weight. Now measure how much amount of dose has been delivered by your meter wall. Then here we can find out that whether it is delivering accurate doses or not. Then net content. Net content of your material or your aerosol container can be calculated just by taking your total weight of your product along with your container, and you have to take the empty container weight, which is used for the filling of your aerosol, and then just minimize or reduce your actual cans or your empty containers weight from your filled container weight, and then you will get the actual content of your total aerosol. Then foam stability. Suppose you are using such kind of aerosols which are having the ability to deliver the foam. So here visually we can inspect your foam stability, whether it is having the long duration of that foam or whether it is having the ability to suppose any mass we are keeping on that foam, whether it is easily penetrating inside the foam or not, at how much time is requiring to insert that object inside the foam. So such kind of we can say physical test we can carry out for that foam. Then particle size. Sometimes suppose we are using the suspension type of your system, and in that we are suppose disperse your drug. So in this case it is necessary to find out the particle size of that drug which is suspended inside the propellant. So in that case we can use the cascade impactor or light scattering device to measure the dose to measure the particle size of your drug. Here cascade impactor is there which can be used, or light scattering device is also used. Then biological testings are there. So after along that all your physical tests, biological tests are also important. Generally therapeutic activity we should know whether your drug is having the ability to do the therapeutic effect or not. So here for the inhalation type of aerosols we have to find out how much amount of dose is required, and then we have to go for the preparation of such kind of inhalation type of aerosols. So by using the animals we can check how much amount of dose is able to give your required therapeutic action. Then for the topical purpose, when we are preparing the topical type of aerosol, here your product is just applied on the affected area or the tested area, and then we have to wait for the absorption of that drug inside your skin, and then we have to note down how much amount of drug is reaches into your blood. That can be calculated by using the blood sample, and with the help of your spectrophotometer, we can find out that how much amount has been absorbed inside your blood. So such kind of tests are carried out for the topical type of aerosols. Then toxicity test. For the inhalation types of aerosols, we have to check the toxicity. Exposing such kind of aerosols for the test animals, we can find out the toxicity. How much amount of dose is having the ability to give the toxicity to that animal, that can be calculated. Then for the topical types of aerosols, we have to use the animals and we have to spray it onto their skin, and we have to find out any kind of irritation is there to that animal. Also we can use the human volunteers, and we can spray that aerosols on your skin, and we can find out any kind of irritation or any kind of chilling effect is there. Such kind of test we can carry out for your topical type of aerosols for its toxicity. So this is about your aerosols.